Unlike your traditional microscope, this one's upside down. So in this case, we're actually looking at things from underneath. The objectives are underneath the stage, and your specimen sits in a dish on top of the stage. The light comes down from the top. We have this chamber around it because we can use this microscope to look at live cells. When we're growing live cells, they need to have certain conditions to keep them happy in order to grow properly. We can also look at fixed cells. Fixed cells are chemically treated to keep the cells in a lifelike state so that we can look at them at different points in their process. We also can use fluorescence. Fluorescence allows us to label the cells or parts of the cells with a very specific molecule, which is going to excite with one wavelength or one color of light and then emit in a different color. And then we capture that second color and use that as part of our imaging process. In this experiment, we're looking at cells that were grown in a multi-well plate, and they're exhibiting this really interesting structure called a neutrophil extracellular trap, or a net. As the name suggests, nets are networks of fibers that our immune cells use to throw out and capture or trap pathogens and other bad things. This microscope is highly automated, and it allows us to go from each well in the dish, take multiple pictures, capture them, and analyze them on the fly.